Okay, so we'll start the class. Try and understand what I told till now about tRNA, because we are going to discuss translation, last step in central dogma of life. Right? Last step only now, replication, transcription, translation. So translation we are going to develop. Then we have seen that since protein structure, the polypeptide sequence and mRNA are completely different from each other, there is no complementarity between the two. There must be something which can read between the two, or you can say there is something which is going to connect the two. For DNA to RNA, for a DNA to RNA thing, complementarity is going to work. Yes or no? Just the complementary sequence. For A you have to put U, for T you have to put A, for G you have to put C, and for C you have to put G. Yes or no? But now from this to a polypeptide, there must be something to link these two, because there is no complementarity here. The two sequences are completely different. We know that there are 20 amino acids which is going to form these polypeptides. Yes or no? There are 20 amino acids, so there must be something which is connecting the two, and we get the answer. What is that thing which connects the two? tRNA. Which connects the two is nothing but a molecule of tRNA called transfer RNA. Earlier, before the discovery of our genetic code, people are knowing about tRNA. That time it is also called as soluble RNA. Also called as soluble RNA. But after the discovery of genetic code, people start saying it's adapter tRNA, adapter molecule, because it's adapting the sequence of mRNA and converting it into a sequence of polypeptide. Yes or no? The actual transfer, the bringing of amino acids is done by tRNA only. Now try and understand what is the sole role of tRNA. Transfer RNA is to carry the amino acids into the growing polypeptide chain. Yes or no? So till now what we have seen, we have three types of RNA now. RNA is going to be of three types. First is mRNA. Second is rRNA. And third is tRNA. These are the three basic types of RNA. Yes or no? Okay. At least you can speak. Other people are not speaking. Reshma, Pujita, Sanmukha, Sheetal.
Yes, sir.
Thank you, Sanmukha. At least you speak. Thank you so much. Try to speak. Otherwise, how I'll feel only I'm speaking in this class in front of a camera. Okay. Okay. Now try and understand here. We have three types of RNA. The mRNA is going to be actually going to code for a polypeptide. So this is the actually coder. The code will be here. This will suggest which amino acid. The sequence of amino acid is in correspondence with the mRNA sequence. rRNA, ribosomal RNA, going to be present in ribosome. Now ribosome means what? They are the actual protein synthesis factory. In case of cell chapter, we have seen the organelle structure and its function. So ribosome is an organelle which function is to do protein synthesis. They are either free in the cytoplasm or get attached with the endoplasmic reticulum. Yes or no? So rRNA will be present in ribosomes. Now ribosome is not just rRNA. Along with rRNA, we have a lot of different proteins to make a ribosome structure. But the component of rRNA will be there. Now, tRNA is the transfer RNA, means what? It's going to bring the amino acid which is present in the cytoplasm to the growing polypeptide chain. That's why it's called tRNA, transfer RNA. Clear? Now try and understand few points. About tRNA. Now if you see this tRNA in a proper sequence, so this is a 3D structure of tRNA, or you can say a tRNA molecule. Earlier, it's going to be like this only. 5 prime, 3 prime. It's a RNA molecule now, called as tRNA. Transcription happened, but not translation happened, and because of that, they are also categorized as genes, or regulatory sequence. Not going to code for a polypeptide, but still have regulatory functions. Now try and understand. In this, at 3 prime, this 3 prime region is called amino acid accepting end. Amino acid end, you can say, or accepting end. This particular region is called as anticodon. Anticodon. Now the beauty of this anticodon is they'll have codes here. They have mRNA. Now it means they have bases, 4 bases, A, U, G, C. So these bases are complementary to the mRNA sequence. So here, whatever the base is going to come, these bases are going to be complementary to the mRNA sequence. So anticodon bases are complementary to mRNA sequence. People can remember this, that anticodon bases are complementary to mRNA sequence. So now understand, if we have this mRNA, right? I told you people that from here only the translation is going to begin now, 5 prime to 3 prime. So these codons present here must be read, and this reading is again in the terms of complementarity, the genetic code, 3 letters at one time. So the 3 words are going to form one code. Remember genetic code characteristic. So now these 3 letters will going to count at once. So we are going to count first 1, 2, 3, 4 and so on. These 3 letters present here have a complementary sequence with the anticodon of tRNA. This much people can remember. Shall we go ahead, everyone?
Yes, sir.
Pakana? Now try and understand. We are saying that for every amino acid, there is a tRNA. For every amino acid, there is a tRNA. We know that there are 20 amino acids, standard amino acids. Then how many different type of tRNA we have in a particular cell? For every amino acid, there will be a tRNA. Because one tRNA can bind with one particular amino acid. It's not like one same tRNA can bind with any amino acid. No, one amino acid is going to bind with one particular tRNA. Now then, if we are having 20 amino acids, how many tRNAs are there?
20.
Right? Okay, now? So we have 20 amino acids for that. It means 20 different amino acids are roaming in the cell cytoplasm. These things are happening in the cell cytoplasm. When there is a need for that particular amino acid, that particular tRNA is going to combine with the particular amino acid and bring it to the growing sequence, growing polypeptide or the protein synthesis factor. This much is clear with you people. The tRNA is clear. Now understand 2 points more important here. The structures are given in the NCERT. Now the 3D structure is given and 2D secondary structure is given. In case of 3D structure, the tRNA will be look like inverted L. The 3D structure of tRNA is what? Inverted L. While 2D structure of tRNA is clover leaf. And this is the picture of clover leaf. This is the picture of clover leaf. This is a picture, this is how it will be look like. Clover leaf is what? Secondary structure. Of tRNA molecule. Right? For 3D structure, what is the shape? Inverted L. Is the structures for tRNA. These 2 points please remember. Okay, important from point of view of examination, NEET examination. These 2 examples are given. Now you can see that this particular tRNA is carrying a serine at 3' end and this particular is carrying a different amino acid at 3' end. Now try and understand what I am saying here. This is the anticodon region and this is the anticodon region of this particular tRNA. And this is the codon of this particular mRNA. This particular anticodon is just complementary to codon, just what I said. Given in NCERT, try and understand A to U, G to C, U to A. Similarly, this A to U, U to A, G to C. Now, what you have to remember here from this can be asked in question form. That they will give what? They will give this sequence. They will give this sequence. We know that AUG is the start point. Yes or no?
Yes, sir.
AUG is the start point for the translation, right? I told last class that it will start from AUG and end at any of the 3 stop codons or nonsense codons AUU, AUG, UGA. Yes or no? Getting idea, everyone? Now, try and understand what question they'll ask. What they ask is for AUG codon, what will be the anticodon? What will be the anticodon? And they gave in 5' to 3' direction. Now try and understand. So for AUG, what will be the anticodon? In case of 5', 3', can you please tell me? For AUG, now we know that the reading sequence will be in this manner, 5' to 3'. The anticodon will be how you are going to write anticodon. So we have to 5', A to U, U to A and G to C and then 3', and just reverse this. It will be UAC 5'. Which one according to you, A or B, is correct for anticodon nature of tRNA?
B.
B is the correct answer, right? A. Ankit is saying A, right?
No, sir, B.
B, okay. Pujita has said, everyone has said B, right? So please remember, dear, they may give a sequence and they ask for anticodon. So you have to take care of these things. Sorry, you have to take care of these things. Pakana, now try and understand what is there. This is the 5', A, C, U. So just opposite. Pakka, this is what is there now. Correct or wrong? Huh? Everyone said B. Correct or wrong? Say bacha, by looking at this is a 5 prime, right? U, A, C, U, and then 3 prime. So 5 prime A, U, C. 5 prime. How come we have written U? One second. Oh, they have given different sequence. Sorry, my fault. Yes, correct, Acha. Done now. My fault. They have given different sequence.
Yes.
We have used different sequence. My fault. Okay, now we will go for translation. Now in translation, what is the actually thing happening? This will have a mRNA, and to that mRNA, or mRNA is going to bind. Sorry, ribosome is going to bind. So this is the sequence of an mRNA, suppose. And I told you there are some parts at both ends which are UTRs. It means untranslated region. So we have to search for AUG, and from where AUG will be there. From there it's going to start, and here must be some stop codon. Either UAA, UAG, UGA. Right? Now try and understand here. At this point, the ribosome is going to bind. Right? And we know that ribosome is consists of 2 parts, bigger subunit and a smaller subunit. Try and understand the first step in the translation process. First step in the translation process is going to be amino acylation of tRNA, which is also called charging of tRNA. Amino acylation of tRNA or charging of tRNA. What happens here is within the cell cytoplasm, when the amino acids are roaming free, right? When amino acids are roaming freely, they will attach with a tRNA. They will attach with a tRNA. And this process is called amino acylation or charging of tRNA. So attachment of a free amino acid to a particular tRNA is called amino acylation, and this is the first step in the translation process. Amino acylation of tRNA, charging of tRNA, and for this process to happen, we have to invest energy in the form of ATP. In the form of ATP will be used here. So for amino acylation or charging of tRNA, which is nothing but attaching a amino acid, a free amino acid to a particular tRNA, is what? Amino acylation process or charging of tRNA, which is done with the help of ATP expenditure. Done? Now try and understand. Second process. Once the amino acid is been attached to a tRNA, what comes next? The attachment of small unit, small ribosome subunit. Plus mRNA. Now the small subunit of ribosome is first attached to a mRNA. First step, charging of tRNA, amino acid is attached to a tRNA. Then the smaller subunit is going to attach to a mRNA which is freely moving or just come out of nucleus for transcription. Done. Now what happens next? To these arrangement, the large subunit is going to add. Clear? First this, second this, smaller subunit plus our mRNA, and to this we are going to add the large subunit. Till this is clear, everyone? Reshma, Ankit, Avani, Pujita, Shanmukha, Sheetal, clear?
Yes, sir.
Super. Now try and understand what happens next. We know that we have triplets, right? I'm just making them far away, but there is no gap here, right? So this is the first one, second, third, fourth one. So first our ribosome is going to bind here at this place. So here we have a larger subunit, and just below this we have a smaller subunit. Smaller means larger. Smaller subunit. This is the larger subunit, this is smaller subunit. And we know that larger subunit has 3 sides. The larger subunit, this one, has 3 sides here. What are the sides? The first site is called as A site. The second site is called as B site. And the third site present here is called E site. We are not going to discuss now E site, just forget about E site, A site and B site. And I told you people, A site is the one which is name come from amino acid site. Amino acid site. P site, the name comes from the peptide bond site. It means what? The growing polypeptide, our growing polypeptide will be held at this particular site. Our growing polypeptide will be held at this particular site. Now try and understand. An amino acid connected to a tRNA, who is going to bring tRNA? Sorry, amino acid, tRNA, right? So a tRNA with an amino acid is going to come to this side. This is moving, so here we have what? A side, here we have B side. So once the first codon is read by our this small subunit mRNA and ribosomal assembly, the first tRNA will be accumulated at A site called amino acid site. Now try and understand. Here we have only amino acid present here. No peptide bond is present. For a peptide bond to be present, we must have 2 amino acids. Now what happened? This whole sequence is going forward at this second amino acid. So what happened? Now this sequence is going to read this particular codon, and for this particular codon there is a tRNA with anticodon complementary to that, having a connection with a tRNA, charged tRNA you can say. So what happens? This particular tRNA is going to come here now. With this, and now this side is free now. So here another tRNA is going to come in accordance with having a complementary sequence with this codon. Now take another color and just draw one more. So here we have one. Now try and understand, we got one more here at A side. This earlier one, the first one, is holding that one amino acid at this side, P side. Now I told you people, the two charged tRNA are so much energetically favored that bringing them closer enough will have the ability to formation of peptide bond. If you are going to bring two charged tRNA molecule close enough, their amino acids are going to make peptide bond. Here we are utilizing a catalytic property of ribosome 23S. Which catalytic property? Ribozyme. So bigger subunit consists of 23S in case of prokaryote, right? And this is going to have ribozyme. And ribozyme is what? An enzymatic activity. Enzyme is going to do what? Increase the rate of reaction without getting involved in the reaction. Yes or no? Biomolecule, enzyme chapter, topic. So what happens here? Now you can see the two molecules are here close enough, or by themselves they are going to form a peptide bond. Or you can say there is the automatic formation of peptide bond. Now what happened next? When this sequence is moved forward, I am going to read the third codon. This side must be free. So what happened next, dear? So next sequence, this having A side, P side and E side. So when the third amino acid is going to come here, at this place, this one will transfer here. So now here we have this amino acid holding what? This amino acid of its own and this amino acid. Connection with the peptide bond, first one. And this is going to go to this exit point. And this is free now, uncharged or not connected with any of the amino acids. Now from exit side, this tRNA is going to exit the complex. And again when next codon is going to come, this is going to come here, and again you will have this particular amino acid attached and the sequence is going ahead. Now we can see that like this only we are moving now. So at the end, first amino acid will be the first codon, second will be the second, third will be the third, like that, so on and so forth, until unless we reach a non-coding sequence. Non-coding sequence, UAA, UAG, UGA. Once that is going to be encountered by this moving complex, what will happen? We know that there is no tRNA for stop codon. There is no tRNA for stop codon. Then what happens? No tRNA will come here, and then a protein in your book written, a protein called releasing factor will attach here, and there will be a termination of this polypeptide synthesis, or you can say translation. This you people understood? This you people understood, dear? Everyone, any problem at this stage? Okay, so the first codon is what? AUG, and for AUG, what is the first? AUG is going to code for methionine. Methionine is an amino acid, so always methionine will be there in case of eukaryote. In case of prokaryote, methionine only, but a little change. It's called N-formyl methionine. For eukaryote, methionine, AUG. In prokaryote, AUG means N-formyl methionine. Always. So we can say that for initiation there is a tRNA, but for termination there is no tRNA. Clear? This you have to remember, and how the sequence is moving, and once how the things are moving. One more thing, the 23S rRNA of bigger subunit is having the property, is having a catalytic property which is called as ribozyme, which is actually playing the role here of increasing the rate of polymerization, this polypeptide growth. With this, I hope you people are done with translation. Clear? This is the picture they have given, so you can very well see that one tRNA is coming in the growing polypeptide chain 5' to 3', right? Like this it is going, and one charged amino tRNA is coming. They are getting attached here, then getting transferred here, and this one is getting away. So A site, P site, and E site, right? And like this complex will go until unless you are going to encounter a stop codon. Done? Done there? Any question, anyone?